Dear students, today we will see the potential targets for antibacterial drugs. There are two targets for antibacterial drugs. First is the biochemical reaction and the second is the form structure of the cell. In biochemical reactions, there are three classes. Class 1 reactions, these are poor targets for antibacterial drugs. Class 2 reactions, these are better targets. And class 3 reactions are important targets for antibacterial drugs. The second major class is the formed structure of the cell. These include the plasma membrane or the cell membrane of the bacteria, microtubule function, or muscle fibers. These three are the formed structures of the cell that can be targeted by antibacterial drugs. So, what are those biochemical reactions that are potential targets for antibacterial drugs? What are those chemical reactions that are included in class 1, class 2 and class 3 reactions? So in class 1 reactions, there is a generation of ATP by utilizing glucose and some alternative carbon source, and synthesis of simple carbon compounds that are used as precursors in next class of reactions. So these are included in class 1 reactions. In coming slide, we will see whether these are useful targets or not. As we have seen earlier, these are not good targets. These are poor targets for antibacterial drugs. But we will see why these are not good targets. Second is the class 2 is utilization of these precursors. The precursors formed in class 1 reaction, these are uh, there is a utilization of these precursors in an energy dependent synthesis of all the amino acids, nucleotides, phospholipids, amino sugars, carbohydrates, growth factors required by the cell for survival and growth. Class 3 reaction is the assembly of small molecules into macromolecules. These include protein synthesis or proteins, RNA, DNA, polysaccharides, and peptidoglycans. So these are in included in class 3 reaction, these synthesis of these macromolecules. We will see which macromolecules are those. Class 1 reactions are not promising targets for two reasons. First is bacterial and human cells use similar mechanisms to obtain energy from glucose. Second, even if glucose oxidation is blocked, many other compounds like amino acids, lactate, etc. can be utilized by bacteria as an alternative source of energy. So these class 1 reactions are not promising or good targets for antibacterial drugs. Class 2 reactions are better targets because some pathway exists in pathogens but not in humans. Cells, there are several examples. So these are better targets as compared to class 1 reactions because the same pathway does not exist in pathogens and human cells. Some pathways are present only in pathogens and that is not present in human. For example, in folate synthesis, in folic acid synthesis, the pathway is found in bacteria but not in humans. Folate is required for DNA synthesis in both bacteria and humans. It is required. So this is the basic requirement of humans and bacteria for DNA synthesis. As in humans it cannot be synthesized, so it must be obtained from diet and concentrated in cells by specific uptake mechanisms. By contrast, most species of bacteria, as well as the asexual form of malarial protozoa, lack these essential transport mechanisms. So, if the transport mechanisms are lacking in these bacteria and in protozoa, for example, in malarial protozoa, so due to deficiency of these transport mechanisms, they cannot utilize the preformed folic acid. Therefore, they cannot use they cannot make use of preformed folate, but they must synthesize this de novo. Sulfonamide contain the sulfonylamide moiety, which is a structural analog of para-aminobenzoic acid or PABA, and This PABA is essential in bacterial synthesis of folate. As we have seen in our previous lectures, how the PABA or how the folic acid synthesis is inhibited with the help of antibacterial agents. For example, sulfamethoxazole and uh, other members of this class. Sulfonamide compete with PABA and thus inhibit bacterial growth without impairing mammalian cell function. So they have minimal effect on, or they have no effect on mammalian cells. So they do not disturb the normal functioning of human cells. The utilization of folate in the form of tetrahydrofolate as a cofactor in thymidylate synthesis is a good example of a pathway where human and bacterial enzymes exhibit a differential sensitivity to chemicals. So these class 2 reactions are better targets as compared to class 1 targets. Protein synthesis. Protein synthesis takes place in the ribosomes. This process we have also seen earlier in our earlier lectures, that how the protein synthesis is uh, going on inside the bacteria. Eukaryotic and prokaryotic ribosomes are different, and this provides the basis for selective antimicrobial action of some antibiotics. Even if we consider the protein synthesis inhibitor drugs, so they behave differently. They act in three different mechanisms, as we have seen in our previous lectures. The bacterial ribosome consists of a 50S subunit and a 30S subunit, whereas in mammalian ribosome the subunits are 60S and 40S. So collectively it forms 70S, and this one will form 80S ribosomal unit. The other element involved in peptide synthesis are messenger RNA, which form the template for protein synthesis, and transfer RNA, which specifically transfer the individual amino acid to the ribosome. As we have seen in protein synthesis inhibitor drugs, so this role of transfer RNA, the role of messenger RNA has been discussed already in detail. The ribosomes has three binding site for tRNA termed as A site, P site and E site. So this transfer RNA brings the new amino acid at A site, and then it moves towards P site and exit through this E site. So in this way the peptide chain is formed, and which is released afterward, and a protein is formed through this process. Nucleic acid synthesis. Nucleic acid synthesis is an essential for gene expression and cell division, interfering with which is an important mechanism of action of many chemotherapeutic drugs. There are 5 different ways of this interference. So we can interfere with nucleic acid synthesis by 5 different ways. First is nucleotide synthesis inhibition. So these are the precursors that are required for the formation of nucleic acid. Nucleotide synthesis inhibition, alteration in base pairing, properties of DNA pattern, DNA and RNA polymerase inhibition, we inhibit this enzyme, DNA gyrase inhibition, and lastly, direct effect on DNA. And this is only in case of anti-cancer drugs, not in case of antibiotics. Third is the inhibition of DNA gyrase. The fluoroquinolones act by inhibiting the DNA gyrase, and these chemotherapeutic agents are particularly useful for treating infections with gram-negative organisms. So the recommended books for this topic are Goodman and Gilman, Manual of Pharmacology and Therapeutics, Rangendale Pharmacology, and Basic and Clinical Pharmacology by Katzen. Thank you very much.